And just distal to it, or proximal to it, there will be this swollen part. We'll be calling it bulbous cordis. And then this will be followed by the primitive ventricle, and then will be the primitive, this venous region that will be forming the atrium. So this tubular structure is what will be forming the heart and a lot of the different, it's arterial and venous offshoots. Let's look into, I will close this again and I have to go back to my image. I will open it as a figure file and I will be making it larger again. Now it's much better. Now let's look what is going on. If you look carefully, this look at this saccular part, this kind of a spherical part, we typically call it bulbous cordis. Remember this name. This name is unfortunately, or this name is important. Now, in bulbous cordis, the process there is you can roughly divide it into three zones. You can divide it in this proximal 1/3. I hope you can see my cursor. You can divide it into the middle 1/3 and then one of this distal 1/3. Proximal 1/3 somewhere here, middle 1/3 somewhere here, and distal 1/3 somewhere here. And these three structures are very, very important because this proximal 1/3, what we will be having here, that will form the right ventricle. This primitive ventricle, what is the region here, that will form the left ventricle. Don't confuse it. This is what students often confuse. They think the primitive ventricle forms both the ventricles. That's not true. Primitive ventricle actually forms the left ventricle in adult heart. This proximal 1/3 of bulbous cordis is what actually forms the right ventricle in adult heart. This middle 1/3, which is sometimes also called as conus or conus cordis, this conus actually forms the outflow tracts of the heart, and this distal 1/3 that forms the kind of the aortic sac. And what happens, or what actually forms the aorta and the arch that we will be noticing very soon. And then comes the primitive atrium, and this is the venous region, and they have two outflow tracts or inflow tracts over here. These tracts are, this is called, sometimes also called as sinus venosus, because this is all about venous blood accumulating here. So we call it sinus venosus. I will not have time to discuss it today because it is at least a couple of hours. But this sinus venosus is most important because on both sides, they are again connected to three embryologic venous system. One is the vitelline vein, another is the umbilical vein, and finally there will be common cardinal veins. And these three veins, our fetal venous structure or embryological venous systems, will give rise to the entire venous system of the body. Now, unfortunately, once again, I don't have time, but I don't know, maybe if we meet at the end of the year or beginning of the next year, I'm very, very happy to go through like a crash course over this vein and how the sinus venosus develops and all these three veins form the different venous system inside the body. Today, we'll leave it over here, and all what you need to know that this venous end will be the primitive atrium, and actually this will form the atria of an adult heart. And now let's come back to this truncus arteriosus. We have seen the proximal 1/3 will form the right ventricle. The conus, the middle 1/3 guy, that will be forming the outflow tract, and then will be the distal 1/3 that will forming the aortic sac. And from the aortic sac, you will be seeing two kind of tubular structures coming over in this arterial end, and they will form the arterial arches, which we will be discussing very, very soon. So overall, this is our main guy in the development of the heart. It is a kind of a symmetrical structure. It's definitely not, the right side and left side are pretty much mirror image to each other through this midline. And these are the different segments of this of the fetal heart, which I think you guys should definitely remember. I will go back to our original picture now. So here comes. So we were so far, I hope you can see the cursor here. Now, we were so far discussing about this. So this is the very important part. Now, what will happen is, now here it gets very exciting. Over time, this 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 primitive cardiac tube will keep on growing and increasing in length. And what you have to realize that this cardiac tube is encased within a pericardial sac, and the space within the pericardial sac is pretty limited. So as this cardiac tube keeps elongating, there is not enough space for it to keep on looking straight or being this linear within the pericardial tube, because there is the pericardial tube and the space inside is pretty limited. So what will happen is eventually there will be now folding of this cardiac tube, endocardial original tube, and that will fold. And now what you need to realize, you have to this fortunately this diagram is color coded. So these blue guys are all the venous blood, the reddish guys are all the arterial blood, and these segments. So keep looking, there will be a folding, and because of this folding now this part, which is the primitive ventricle, that will come towards the left because of this folding. This proximal 1/3 of the bulbous cordis, which were forming the right ventricle, that will come to the right. So I hope you can see the primitive ventricle will finally end up on the left. That will form the left ventricle. The proximal 1/3 of bulbous cordis will come towards the right. That will form the right ventricle. Now here comes the interesting bit. Look into these blue guys, which is the sinus venosus or the venous end. What will happen is once these ventricles are established with development, this venous end will keep folding from the back, and they will come over on the back side, or shall I say the dorsal side, and they will form the right atrium and left atrium. And most of these veins, especially on the right side, the cardinal veins, the vitelline veins, and uterine veins, they will disappear, especially on the right side. On the left side, they will be there, and that will be forming different structures, but we will go back into that in later. So bottom line is, what you need to understand is these endocardial tubes, they fuse, it forms a kind of a symmetrical linear structure, but because there is not enough space also within the developing embryo plus the pericardial cavity, it has to fold. And because of this folding, this primitive ventricle comes to the left side, forming the left ventricle. The proximal 1/3 of bulbous cordis comes to the right side, forming the right ventricle. And this entire venous end keeps folding, and it gets folded on it from the dorsal side. And thus you have the right atrium and left atrium. And that is where the venous blood will come over. Now, so what we actually see, this is where it starts looking like a proper heart that we know in vertebrate systems. Now, here is another thing. We were discussing about the two tubes here. So this is the arterial end. There were two tubes over here, and we can look over. And these two tubes actually form the aortic arches. These aortic arches will be important, and we will be talking about it in a moment. So we have seen now, for example, and for your knowledge, this figure is very, very important. I have already shared it with Armanda, and I don't know, I'm pretty sure he has shared it with you. If not, he will share it with you after my talk. Remember, this is a fantastic image, and that actually shows why the heart is so convoluted, and it is a folded structure like the way we know it now. It starts from very primitive stage at here, but it ends up over here. And we'll look into some of the other development structures or developmental stages in heart in next few moments. Now, what I also want to talk about today are some of the pharyngeal arches, because we ended up in the aortic arch. So I will again go back here, and that will make it clear. So we have discussed the development of heart over here, and now let's look into these aortic arches. Now, aortic arches, what have you heard of pharyngeal arches? I mean, Armanda, do you guys teach pharyngeal arches to students? Hello?
Yes, I've already said it to the students.
Very good. So that already makes my life much easier. So I will be now sharing, I hope you can see that. I will be starting the laser pointer. So this is kind of the embryo, a vertebrate embryo, and doesn't matter which species, because fortunately the vertebrate embryos, at least from outside, external view, they look pretty much the same. Doesn't matter which embryo. Usually this looks very, very similar. And what happens is with the development, as the embryo keeps developing, it gradually attains kind of a cylindrical shape. As you can see, this is a cylindrical shape. We are looking on a left lateral perspective. This is on 4 weeks, 5 weeks, 6 weeks. Let's not get into this because this timeline keeps changing with the species. That's not where we are interested because that's pure information. We are interested in understanding the overall anatomy and how it develops. So this is typically kind of this. And what the interesting bit is, if you follow my cursor, this is where the bulge appears, and this is the bulge where the heart, fetal heart is developing inside it. And you can see over the times the different somites have appeared. The otic plateau, so the eyes will be developing over here, and you can see gradually the limb buds will keep developing. That's not we are interested. We'll straightway go into the pharyngeal arches. I'll not spend a lot of time because Armanda told that we have been already taught, or at least the introduction to pharyngeal arches were made to you already. Now, the interesting bit of pharyngeal arch, and I keep on telling to my students that always try to learn the development of head and neck systems starting from pharyngeal arches. The pharyngeal arches, you might ask that if it is about head and neck, how does all these cardiovascular system? The interesting bit is if you look into the vertebrate pharyngeal arches or the pharyngeal apparatus, there are usually 6 pharyngeal arches. 1, 2, 3, 4, 5. Now, out of these 6 pharyngeal arches, the number 5 usually digresses, so it disappears. Disappearance is not probably a perfect good word, but it definitely digresses. It doesn't exist as such in adult animals. We still don't know why. It is one of the fallacies that the 5th pharyngeal arch basically does not develop, it digresses. Now this is, so we are, if you come over to this figure, you will see we are basically remaining with pharyngeal arch 1, 2, 3, 4, and 6. Number 5 somewhere disappears. Now, for each pharyngeal arch, you will have some kind of a kind of artery, a vein, and a nerve. And these are and also a cartilage. This is very important also because if you understand these pharyngeal arches and what are the structures they develop, as students struggle to remember all the structures and their innervation and the arterial supply when learning neuroanatomy. But pharyngeal arches give you that wonderful logic which makes you realize why especially our head and neck systems look the way they are. Because most of our face and our plate and our what we typically call as the cranial part of our body comes from the pharyngeal arches. Now, I will focus on, so this is kind of a more complex structure of the pharyngeal arches. This is the lateral, and you can see the heart has already developed. And this is a kind of a more lateral oblique purview showing the different pharyngeal arches. This is so you can see the first arch, second arch, third arch, fourth arch, and the fifth arch. This is where the neural tube will develop. Now, why am I interested in pharyngeal arches? Because all these pharyngeal arches will produce substantive structures. And let me show you this. I have also shared this material with Armanda, and I'm pretty sure he has shared it with you. If not, he will definitely do that, I'm 100% certain. So these are the different derivatives of the pharyngeal arches, and you can use it as your teaching material as well. I will not go into detail because that's not what my aim is. I'm trying to make a different point, but first let's go through these 5 pharyngeal arches. Remember, number 5 is missing, so that's not written here. So there is no number 5. It's only 1, 2, 3, 4, and 6. All these pharyngeal arches will have its own artery, nerve, muscles, and bones. And we are interested in the arterial part because that is part of the cardiovascular system. Remember, these aortic arches, there are altogether 6 aortic arches. These aortic arches, 6 aortic arches are contained within the 6 pharyngeal arches. And as I said, the number 5 disappears, or shall I say digresses. So the number 5 is not mentioned over here. We are interested in the arterial segments that are coming from the aortic arches, and that is where it will get very, very exciting because fundamentally the cardiovascular system, especially the arterial part, is coming from these pharyngeal arches and the different branches of aortic arches that are contained within this pharyngeal arch. So without wasting time, I will be now going to show you the different pharyngeal arches and how it forms. So first, let us go through a complete view of these aortic arches, because these are the aortic arches. Let's look into this. So this is where the heart is, heart tube that we were discussing. But we are not showing in the heart tube now because we want to see the entire structure. So this is where the heart is, the endocardial tube, and is over here, and they split for these 5 branches, and these 5 branches are for 5 pharyngeal arches. Remember, so this is number 1, number 2, number 3, number 4, number 5 is missing, and number 6. This is the ventral part, and this is the dorsal part. You have to understand this is a 3D. So this is the ventral, this is the dorsal part of the embryo, and somewhere between them will be the gut, primordial gut developing. So these are the 5 arches. 1, 2, 3, 4, 6, 5 gone missing. Now, what will happen is over time, a lot of these arches will kind of remaneuver and they will become different. Let us see if we can get through this. So for example, this is another view of these aortic arches. So each arch is now contained within an individual pharyngeal arch. And to understand it well, I have also shown the 5th arch, which will gradually disappear. So this is the original kind of the system of the aortic arches within the embryo. But, so this is the right first arch, this is the left first arch, second arch, right and left, third arch, right and left, fourth arch, right and left, fifth arch, right and left, sixth arch, right and left. This 5th arch will disappear. As we were discussing, the 5th arch, it originally develops, but then it disappears. And then remember 2 very important structures, the intersegmental arteries. These intersegmental arteries are the primitive arteries that develops, and basically they form the different, they supply the different somatic structures of the developing embryo. Now, these intersegmental arteries, they basically form in an adult animal, they form the intercostal arteries of thorax and the different other segments. Let's not get into there. But for the development of cardiovascular system and the aortic arches, you have to remember the intersegmental artery right and left, the 7th one, because the 7th intersegmental artery right and left, they form important structures, as we will note it now in few slides. And on the middle, this is the aortic sac, ventral sac. So this is basically where this original tube that we were discussing that were here. So that's where we will begin with. Now, this structure, these are arches of aorta, that definitely does not look like adult animals. Because adult animals, if you look into this colored part, that is how the aortic arch look. But it started a very symmetrical structure, but ended up in an asymmetry, which then technically develops. And this is where it is interesting, how does it develop? Because this is how original structure of the design was, and we ended up in a design like this. How does it develop? And what is one of the important things is, so let's see how it develops. And what I will be doing is, I will be opening this figure. So this is the figure, and we want to know how it ended up there. Now, the first thing is, if this is the original design, what will happen is, I'll start my eraser. What will happen is, the first and the second aortic arch, both on right and left, they will simply disappear. So I hope you can see my eraser. I'm erasing it because they will just plain and simple disappear. Here you go. I'm just disabling. So first and second arch, aortic arch, they will disappear. Now, disappearance again is not the proper word. The first aortic arch sometimes stays as a maxillary artery. You might have heard the name. And the second arch that or sometimes stays as the stapedial artery for middle ear. So there are remnants of the first and second arch, but by and large, they will disappear. So now we are remaining with the 3, 4, 5, and 6. Now, we have already discussed the number 5 arch, aortic arch, both on right and left will disappear. So let's also delete it or erase them. Am I clear here up to this point? Armanda, can you see that I'm
Yes,
Yes,
we see.
so it gets much easier now. The first arch, the second arch, by and large gone. The fifth arch gone. So the original complex design is now becoming way simpler. Now let's see what happens. If you look carefully over here, now the third arch and fourth arch, there is a connection here and here. That is usually called caroticus aorticus. What will happen is they will be also deleted here. Here. Now it gets easier. So now we have this one complete arterial system on each side. And this arterial system will become internal carotid artery. Internal carotid artery. So the first arch, second arch disappeared, although not completely. They stay as maxillary artery and stapedial artery. These are not major arteries, but of course they're important. And the third arch, they will form the internal carotid artery. They will get separated from the fourth arch, and they will become now independent. And these are the internal carotid arteries. Now, while the internal carotid artery is developing, there will be a kind of a bud coming over here. I hope you can see. I'm drawing some structures here. Can you see? I hope you, so it's there. Some buds will keep on coming over there, and that will grow, and they will form the external carotid artery. Very, very important, external carotid artery. So this is how the internal carotid system and the external carotid system develops. So if you are asked in exam, and when we were students, we were asked many, many times, which is the aortic arch the internal carotid artery develops? Your answer is the third arch. Now, so this has already developed. Now we are remaining with all this. What will happen now is this I will probably, maybe I should go back here, and this will be now interesting. Just a second. Yeah. So what I already deleted or erased can be seen over here. You can see the first and second has already deleted. This caroticus aorticus has also been deleted. So this becomes whatever remaining is the internal carotid. And from here, I showed you two buds appear. They form the external carotid. Now let's go to, so this was number one. It digresses. Number two digresses. Number three on each side forms the internal carotid. And this budding that comes out, that forms the external carotid. Now let's look what happens here. So what happens here is number four, that is the number fourth arch on each side. Now look, if this is the intersegmental artery. This intersegmental artery and this fourth arch, they will team up, and that will form the right subclavian artery. This is important. The right fourth arch and the right seventh intersegmental artery, they will come together and they will form the right subclavian artery. This will not happen on the left. On the left, it will be only the fourth arch that will be forming the left this subclavian artery over here. You can see here. Again, we'll discuss it, or we will come back to it in a moment. You can see the fifth arch, right and left, they have completely digressed. So we have the internal carotid system, external carotid system, the right subclavian artery, and the left subclavian artery coming over here. The fifth guy on both sides, the fifth aortic arch, they have disappeared. What will be remaining is that now let's deal with the sixth arch. The sixth arch, please remember for exam, because they have been asked many, many times in exam. What is the sixth arch gives rise to? And the sixth arch gives rise to the trunk of pulmonary artery. Remember this, it has been asked many a time. The sixth guy, they will give rise to the pulmonary trunk. And this is interesting because the pulmonary trunk only so let's look on the right side here. So we are discussing the right side. In the right side, if you look into the sixth aortic arch, most of it disappears. Only a small trunk appears over here in this angle, and that forms the right pulmonary artery. And on the left side, this left pulmonary artery appears over here, while most of the sixth arch, left sixth arch disappears, although it remains patent in embryological stage. Remember, 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 this is what we call as ductus arteriosus. I'm pretty sure you have heard it if you're discussing fetal circulation, but ductus arteriosus is very important in fetal blood supply. We'll see that in a moment. Because ductus arteriosus is it only works as an arterial conduit in embryological states, but in adult animals, this will close down. So this is how the six aortic arches come over. The first and second on both sides, they will disappear. Although they will not completely disappear. They stay often as the maxillary artery, and the second guy will be the stapedial artery. After that, the third arch will give rise to the carotid, and towards the midline, the external carotid systems will be attached to it. Now let's come to the fourth arch. The right fourth arch, aortic arch, in collaboration with the right seventh intersegmental artery, will form the right subclavian artery. On the left side, the left fourth arch will form this entire arch, and sorry, the subclavian artery over here. The fifth arch on both sides will disappear. The sixth arch on the right side completely disappears, and then comes a small offshoot out of it that forms the right pulmonary artery. The sixth arch also, a lot of things disappear, and a conduit comes out in this corner. That will be the left pulmonary artery, and whatever is remaining over here, that is called as the ductus arteriosus. This is very important because you have to understand this ductus arteriosus keeps a connection open between the pulmonary artery or the pulmonary trunk and the aorta. Rest, everything will disappear. So whatever you see is in white, that will completely disappear. So from here is the magic of nature that we end up over here. And now you have the complete aortic arch like what we see in adult animals. You have to also remember this right side, where I'm putting my cursor, that will also disappear over development. So now we have this asymmetric structure of the aorta that we know in adult animals. It is very important you remember these aortic arches, because not only the heart, it gives you the structures and understanding what is the carotid systems, why this aortic arch comes like this, why it is asymmetric. Because now what happens is it comes together. You can see the internal carotid, external carotid, and the right subclavian. They come out as the one single offshoot, and that is called as the brachiocephalic trunk. On the left side, you will find this common carotid, which will be splitting as external and internal carotid, and then you have the subclavian. This is how the aorta, the arch of aorta in adult animal develops. And then this is the pulmonary trunk. This pulmonary trunk will be connected in embryos or in fetus to this aortic arch through this ductus arteriosus. Remember this name. This has been asked many a time in the past in medical exams. I will move on. So we have the aortic arches. So this is finally we will end up over here. So this is where we began. Lot of arches got disappeared, blah blah blah. A lot of remodeling happened, and we ended up in the common kind of a thing that we know for sure. This is the structure of aortic arch that we know in adult animals, isn't it? We are not aware of this. We end up here, but that all began here. Slowly things got maneuvered, manipulated, restructured, redesigned, and don't ask me how this all happens. Nobody knows. These are just simple magic of nature. It happens seamlessly. That is why we all exist, we all survive, and most of the times live a happy, productive life. This happens, and this is a fantastic diagram because from here you can see the brachiocephalic trunk splitting into subclavian, external, internal carotids. Then you have the common carotid, and that common carotid splits into external, internal carotid. and then you have the left subclavian. So this is from this very symmetrical structure, a very productive but asymmetric structure develops. And this is where the ductus arteriosus remain. This ductus arteriosus only remains in fetus. Once the baby is developed, baby is delivered, or the offspring is delivered, it closes down and it stays as a ligament called ligamentum arteriosum. We will note that in few seconds, in next few slides. One last thing in this image. So we were talking about pharyngeal arches because these are all aortic arches, but they are all in pharyngeal clefts. So they will be accompanied not only with arteries, there will be also cartilages, there will be also nerves. Each pharyngeal arch has its own nerve. I showed you a structure, a drawing, so that is here. Let me come back here. So this is the PDF we have been with you. It's a very nice. I share it with my students. They find it very helpful for remembering things in the exam. So these are the different nerves that also supply these different pharyngeal arches. For example, pharyngeal arch 1, it will be supplied by trigeminal. So all the structures that develop from pharyngeal arch 1 are supplied by trigeminal nerve, cranial nerve 5. Similarly, the second pharyngeal arch will be supplied by facial nerve. Usually that is the cranial nerve 7. The third pharyngeal arch by glossopharyngeal, cranial nerve 9, and the fourth and sixth, because fifth is missing, they will be by the vagus nerve. Now the vagus nerve, it definitely supplies the fourth pharyngeal arch, but in the what happens in the sixth pharyngeal arch, the branch of the vagus nerve, I will go back to that figure again, hold on for a sec. So here I am opening this. So the sixth pharyngeal arch and the branch of the vagus nerve over here, because the fifth guy disappears, it rebounds back on both sides, and we typically call as, because they are going back, they are rebounding, we call them recurrent laryngeal. It's a recurrent nerve, and they are called laryngeal nerve because this part of the vagus nerve, they keep supplying the extrinsic and intrinsic muscles of the larynx. So if you understand the development of how this cardiovascular system and aortic arches grow up, anatomy becomes much easier to remember or to recall for exam. If you keep trying to memorize all this information, it's a sure recipe for failure in exam or disappointments. Because anatomy, anything that you want to remember or recall should have a logic, and the logic comes from doing it. Very important, I also suggest, because these figures have been all shared with you, it will be very important that you also practice. You draw yourself, you delete these aortic arches as they go, because it's very interesting. From a very symmetrical structure, it ends up in an asymmetric structure, and how the nerves and everything come into relation with it. That is why I keep telling to my students, try to learn things, especially on the cranial part of the embryo, from the pharyngeal arches. These documents have all been shared with you, so I hope it's not a problem. Now let me close it. Yes. So we have seen how these heart arches develop, how they different form the structures, and how they are related to some of the nerves. Now let's get back to some of the other guys. We left our discussion over here. I think we have seen the structure, the endocardial tube, in the right ventricle. This is the bulbus cordis. This is where these were the original arches, and they will slowly form the aortic arch segment, what we have seen. So that's not interesting, or that's not what we are discussing here. Now what we will do is we will focus over this part, the bulbus cordis, because the bulbus cordis is the original, and in fact the middle third of the bulbus cordis is the outflow tract. That means they will form the aorticopulmonary system. So the aorta and pulmonary tract, they both will develop from the same tubular structure. And this is again super, super exciting. And it's a mystery how this all happens, but it happens. So if this is the tubular structure over here in the bulbus cordis, so this is the tubular structure. This is a very nice diagram. The red arrow will typically show you the arterial blood or oxygenated blood, and the blue arrow will show you the deoxygenated blood. So it starts over from here, and what will happen is there will be a septum developing that will divide this single tube into two separate conduits, one for the oxygenated blood another for the deoxygenated blood. Now, here is the mystery. I have told you in the beginning of this lecture that the heart and the cardiovascular system mostly develops from mesoderm. The interesting bit is this septum actually develops from the ectoderm. And a lot of ectodermal cells migrate from the central axis, and it somehow, don't ask me how, because I don't know. And I guess nobody knows. But some of the ectodermal cells will migrate over here, and they will form this aorticopulmonary septum. So that means it will separate, it will distinguish this single tube into two separate subtubes, one for the oxygenated blood, another for the deoxygenated blood. This is not where only the magic ends. This is one of the very few septum known to me that is spiral. It is not a straight cut septum. It spirals. And again, you should not ask me how. It all happens by magic. So if you look over here, when it is starting the original within the parenchyma of the actual heart, the two oxygenated and the deoxygenated conduits, they were lying side by side. But because it's a spiral septum, by the time it reaches over here and the arch of aorta, this is the right brachiocephalic trunk, this is the common carotid, and this is the left subclavian, which is not shown completely, of course, because we want to have a clear view over here. By this time, this structure, they were not side by side anymore, and the pulmonary trunk comes ventral to the aortic arch, and the aortic arch kind of wraps around it from ventral to dorsal. And this is how the pulmonary trunk is here, splitted into two. If you remember, this is coming from the original sixth aortic arch on the right, and this is the left sixth aortic arch on the left. And this is where the ductus venosus somewhere will be over here. And sorry, there will be a connection over here. That will be the ductus arteriosus. So this is a spiral case. Don't ask me how this all happens. But what happens is one outflow tract, there will be an aorticopulmonary septum that will be originating mostly from the ectoderm for a change, and that will, from side by side, it will make into a ventrodorsal orientation of these two tracts. And that's the real deal, because I think this is incredible anatomy, and only nature can do such magic. That's how we have to explain. So we have already discussed how the heart develops. So this we developed. We have understood, or at least tried to understood, their relation with the pharyngeal arches, because every pharyngeal arch contains one of these aortic arches. Now these aortic arches then form and gets remodeled and remaneuvered, and so typically again aortic arches, they will be remodeled again. So from a very symmetric structure, we will end up in an asymmetric structure that we know of, and finally we will end up over here. And now not only the heart has developed, but also the aortic arches have designed. So we have this developed aortic arch as we know, or as we've seen, or as we have drawn, and the pulmonary trunk has developed because of this aorticopulmonary septum. It's the same trunk divided into two halves, which are spiral. And that is the aorticopulmonary septum. We have discussed most of the external bits, but what is going on inside the heart? Inside the heart, especially in advanced vertebrates, the atrium and the ventricles, they're completely separated. We know that. There are variations. Different species have different variation, and this separation is not always complete in every species. But by and large, there is a septum in advanced vertebrates, and the heart tries to keep the deoxygenated and oxygenated blood separate. There are exceptions, but overall, this is our template. Now, what actually happens is, and this is important, so this is the primitive atrium on both sides, and this is the primitive ventricle. What happens is, if you look carefully, there is something called endocardial cushion. These are typically developing some gelatinous structures that develops over here, some on the ventral side, and this is on the dorsal side. They will develop over here. So this is very important that you realize of these endocardial cushions. Once these cushions develop, there are two, you can see there are two conduits, two kind of openings between atrium to ventricle. They are called atrioventricular canals. What is very important that you realize that from this top part, there will be a septum developing over here. I hope you can see the cursor. This is called the septum primum. And the septum primum that will finally come over, go down and attach to this endocardial cushion. Again, these are very complex structures, but it happens like magic. And I sometimes find it incredible how this all happens. So the septum primum, it develops from the top, it goes down and touches the endocardial cushion. But by the time it touches the endocardial cushion, some of the cells on its top part, they apoptotize and they are basically degenerated. As a result, what happens? There is an opening in the septum primum. Now, it's a bit simplification, because by the time this septum primum has this hole inside it, because some of the cells have typically degenerated due to apoptosis, another septum develops over here. I hope you can see I'm moving the cursor on top of it. This is called septum secundum. So what happens is there is a septum secundum, there is a septum primum, but part of the septum primum has disappeared, so leaving a kind of a hole or opening. And what happens is this is the right atrium, this is the left atrium. Now blood can kind of flow from the right to the left atrium, and these two valves, you can see these two leaves or these two parts of the septa, this is septum primum, this is septum secundum, they kind of act as a valve. And this foramen through which the blood flows from right to left atrium is called foramen ovale or oval. Please remember it because we will need it in next few minutes. Foramen ovale will be coming back in next few minutes. Now, these are the two ventricles developing, and this is where the right ventricular septum will also develop. This endocardial cushion will also give rise to the valves. On the right side, there will be tricuspid valve. On the left side, there will be mitral valve, sometimes also called as bicuspid valve. These valves will basically regulate how the blood flow from right atrium to right ventricle and from left atrium to left ventricle. So all this, what we have been discussing, development of the aorta, development of disappearance of arches, development of the pulmonary trunk, they all go through simultaneously. And I hope it's pretty clear that it's really, really complex systems that's going on. So let's look a bit closely to this another segment. So this is where we will discuss how the septum develops. So we have been discussing this figure. Let's look a bit more carefully. So this is the endocardial cushion, and from the top, the septum primum will develop. First, it will develop, it will keep growing, and then initially there will be a hole here that will be called as ostium primum. This hole, by the time this ostium primum is reaching this endocardial cushion, some of the cells on the top, they will go apoptosis, and that will create a kind of a hole. By that time, there is another septum developing on the right to septum primum, that is the septum secundum. This septum secundum will grow, one comes down to up to this layer, and that will reach, they will overlap this ostium primum and ostium secundum. The terminals will overlap. Don't ask me how they all understand it, how much to grow. These are all magic systems. And this will be the foramen ovale, where the blood will flow from right atrium to the left atrium. The ventricular septum is rather simple. That will develop over here, and the ventricular septum from the bottom, it will grow towards the endocardial cushion, and they will attach. Now, when it attaches, so the part that develops from here, that is the muscular portion, and where it makes the final contact with the endocardial cushion, that is the membranous portion. And this endocardial cushion will again, it will differentiate, and that will, as we have seen, will form the different structures, including the very important valves. Those valves will maintain they will regulate that the blood only flows from atrium to ventricular and they don't go back. So the blood flow through the heart is unidirectional in normal cases. If it is not, that can create a lot of issues. So the blood flow in adult advanced vertebrates is unidirectional. It flows from atrium to ventricle and not back. And this unidirectional flow is maintained and regulated by these two valves that will be coming from this endocardial cushion, which started over here. you can see it starts as a smaller structure, but we develop, it really bulges up and produces these wonderful structures of heart, including the valve. And now the blood flow system is maintained and regulated. With this, I will come to the final part, that is the fetal circulation. I hope, Armanda, how much time I am left with? Hello? Hello?
Yes.
Yeah, how
Hello.
much
Yes.
time, yeah, how much time I'm left with? I will finish it in next 15 minutes.
Around
Okay.
10 to 15 minutes.
Yeah, I will finish it in next 15 minutes. Good.
Yes.
Now, so let's talk about the fetal circulation. Now, why fetal circulation is so important? Fetal circulation is important because fetus is completely dependent on the maternal unit. Fetus is dependent on the mother or the pregnant mother for nutrition, for gaseous exchange, and everything. And fetus is not, because it is not an independent unit, it has certain amazing facts. And one of the things is the circulation. Now, fetus is a developing part. It's a fast developing system, so it needs a lot of oxygen. Now, the first question is, the maternal blood is already supplying or getting through the fetal unit. So how does the fetus can extract that extra oxygen? And the answer to that is, again, a magic of nature. The mechanism is fetus has a different hemoglobin. We know, I think it's very, very clear that hemoglobin is the key protein that carries oxygen into the blood. That is the premium carrier of oxygen in blood. Now, what happens is, it is very interesting. Hemoglobin, which is an iron-dependent protein, or iron, and it has a globin and heme part, of course, it also has a lot of variance. So the hemoglobin that we have in our body system now is very different than the fetus has. And this is again a magic of nature, that fetal hemoglobin has much more affinity towards oxygen than adult hemoglobin, which in other terms, fetal hemoglobin binds to oxygen much higher than the hemoglobin that we have as adult animals. This is a magic, because my PhD is in chemistry, and the hemoglobin protein, which is in fetus and in adult animals, that molecule is just slightly tweaked, but that already gives it that special capability that it can extract oxygen. So the fetal hemoglobin, because it has higher binding capability to oxygen, it kind of extracts the oxygen out of the fetus blood and supplies the oxygen to the developing fetal tissues. So remember this, one of the first thing incredible about fetal circulation is fetal hemoglobin. I cannot go to a lot of details about fetal hemoglobin. That's not part of this embryology lecture, but please recall it, and when you go back, please give it a study, give it a look, give it a search, and see what you get. It's an incredible mechanism. There are some other issues as well. Let's look into this image, okay? Let's go one by one. Can I start? No, I cannot start over here. Okay, I have something. Okay, so let's look into this fetal circulation. I can search it here. Hold on. Just a second. Yeah, so this is, you can see my laser pointer. That's pretty good. So this is the umbilicus. This is on the left side. Let's assume this is the maternal unit, and this is the fetal heart. So this is the fetal side. The blood from the maternal side is entering the fetal circulation through this umbilicus. The placenta, original placenta, is somewhere situated here. So the fetus blood, it enters through here. That is number B, it's mentioned here, that is called umbilical vein. Number A is the umbilicus. This is where the umbilical vein is entering, and that gets in. Now, remember two things. One of the issues with fetal circulation is that there are two very important organs developing in fetus that are very solid. One is liver and another is the lungs. The lungs that we know in adult animals is completely different than what it is in fetus. If I ask you how the lungs look in adult animals, we have a very clear-cut view how the lungs look. They are black, they are usually one on each side, they have multiple lobes, etc., etc., etc. In case of fetus, that's not the case. In case of fetus, these lungs are completely not expanded, so they look like a fist, a kind of a solid structure. And on top of that, their color is pink. Remember, the original color of lungs is not black, it's pink. It becomes black because of deposition of carbon particles, because as we keep on respiring and inspiring new air or breathing, we keep on taking trillions and trillions of carbon particles. And those carbon particles deposit on adult lungs to give it that black color. But originally, lungs is pink. And because the fetus is not breathing independently, their lungs are still pink, and B, it's a kind of a pulp. It's not expanded like the lungs we know in adult animals. It's a solid pulp-like structure. So now there is a problem. And the problem is we have two structures in fetus which are much more solid. One is the developing liver. I think we all have seen liver, and liver is a very tough structure. The second problem is lungs. It is also a solid structure. So because these two are solid structures, it is very difficult to push blood through it. Imagine you have a solid ball and you have attached to a syringe and you are trying to inject water through it. I hope it's very clear. It will give a lot of back resistance and you have to put a lot of force to put the water or any fluid through it. That is a problem because the maternal heart is somewhere here and it is pumping the blood that is entering the fetal unit. Now, if the maternal heart has to pump the blood through liver and through lungs, which are both solid, there will be a lot of resistance in this blood flow. And the maternal heart has to pump very hard, which is impossible to sustain. And the maternal heart will ultimately fail. So that's a disaster. Now, nature has been very intelligent to come up with a solution. And the solution is the maternal blood that enters the fetal circle, they bypass these two structures mostly. By bypassing it means most of the blood that enters fetus, they don't go through the real matrix of the liver or the lungs. They bypass it. There is a kind of a detour. And this is the heart of fetal circulation. This is the incredibility of fetal circulation. You have hemoglobin F, which extracts all the oxygen out of the fetal, out of the maternal blood, because the maternal blood has hemoglobin adult. The fetus has hemoglobin fetus. So HbF extracts all the oxygen, plus the circulation in fetus bypasses the liver and the lungs, because these two are solid organs. So to push the blood going through them will create a lot of workload for the maternal heart. Ultimately, it will fail. That cannot be allowed, of course. So here is the magic that happens. The blood comes over here. This is the umbilical vein written here. It goes. Remember, by this time, the fetal liver is also receiving blood from caudal vena cava. This is the part. Number C. This figure is all coded. So all the alphabetical codes are mentioned over here. But most of the blood that is coming from the maternal side, that does not enter the liver. It does not. It flows through a special venous connection on top, on the surface of the liver. And that is called the ductus venosus. Number D. Remember this. Ductus venosus. Don't forget it. I have a question. Okay. Oh, Armanda has left. No problem. So ductus venosus. This ductus venosus is the first detour, or shall we say shunt. This is typically called as the shunts of fetal circulation. The first shunt is ductus venosus. It shunts, it skips the liver. Good, let's follow. So the blood comes over here, and the vena cava is coming from here. The vena cava passes through the liver. We don't have time today, but we can discuss it in next phases. They will all come to this superior vena cava. They will all end up in this inferior vena cava, and they will end up in this right atrium. This is very important to know. This is the right atrium. So all the venous blood and the maternal blood, they will mix up, and the mixing can happen because fetus have hemoglobin fetus. So mixing is not a problem. In adult animals, mixing is a problem. We cannot mix oxygenated and deoxygenated blood. But there are species variations though. But in fetus, mixing is no problem because hemoglobin F is in action. It will extract the oxygen. So the blood will end up in right atrium, and also some of the venous blood will come through the superior vena cava or cranial vena cava, whatever, and they will all mix up in right atrium. From the right atrium, remember, there is a foramen ovale. We discussed it right now, a few minutes back. This is number E, foramen ovale. So most of the blood will straightway move from right atrium to the left atrium. Most of the blood. It will move from the right to the left atrium. And once it enters the left atrium, it will immediately go to the left ventricle, and from left ventricle, it will go to the aortic arch. Depending on species, but usually it is something like 80%. So whatever blood is accumulated here, roughly 80% will immediately be shunted towards the left atrium, and they will follow the left atrium, follow left ventricle, followed by the aortic arch. So it's already in the arterial system. Roughly 20% of the blood goes to the right atrium. So not all the blood immediately gets shunted towards the left compartment. It also comes to the right ventricle. Little bit. This right ventricle, this blood will enter the pulmonary trunk. But there is a problem. And the problem is it cannot go to the lungs because the lungs are not functioning yet. They are solid organs. So there is no point in going to the lungs. They are dysfunctional. So what will happen is, recall ductus arteriosus. That is the connection from left sixth aortic arch. That will use this ductus arteriosus, number F. And that 20%, whatever blood is remaining to the right side, they will straightway access the aortic arch. So in this way, the entire blood that is coming to the atrial system of the heart gets directly moved towards the aortic arch, and the lungs is completely bypassed. So there are 3 such shunts. Number 1, remember this very, very clearly. Number 1, ductus venosus, to bypass the liver. So to bypass liver, 1 shunt, ductus venosus. To bypass lungs, there are 2 shunts, foramen ovale and ductus arteriosus. This ductus arteriosus is coming from the left sixth aortic arch. So in embryology, this question has been asked many times as MCQ. Now, so this ends up over here. So the 100% of the blood gets into the aortic system with nothing for lungs. But it means in the heart, this is the last part of my lecture, there is a mixing of aortic oxygenated and deoxygenated blood, which is okay in fetus because fetus has fetal hemoglobin. But it's not okay in us, in adult animals. Because in adult animals, there is hemoglobin A. So oxygenated and deoxygenated blood needs to be separated. And that is why what happens when the baby is born or the offspring has been delivered, these shunts has to stop. These shunts are not required because the baby is now living as an independent entity. Secondly, these are not compatible with life sometimes because they are mixing oxygenated and deoxygenated blood. So these three shunts will stop immediately or in less than a month, usually, after the offspring is delivered or the parturition has happened. How? Once the baby is delivered, this ductus venosus will disappear or it will digress. The foramen ovale will close. It will close because the moment the baby is delivered and it starts breathing, the lungs, which were like a solid pulp, it's like a balloon. The moment the baby starts breathing, it expands. And the moment it expands, the pulmonary trunk is now functional because now it can divert blood to the lungs. Lungs is functional. And the moment it is functional, this ductus arteriosus is no need anymore. In fact, it's a problem. So this ductus arteriosus will automatically close. And finally, when the lung starts working, now initially what happened, the pressure in the right atrium was higher than the left atrium. So the blood pressure here in the right atrium was higher in the left than the left atrium. So the blood could flow from the right to left side in fetus. The moment the lung starts working, the pressure on the left side becomes higher than the right. So the pressure is reversed. And as a result, this foramen ovale, this valve, snaps down. As a result, foramen ovale is also closed. Bottom line is, these three shunts, ductus venosus, foramen ovale, and ductus arteriosus, they were only there for the fetal circulation. The moment the baby is delivered and it starts breathing independently, these three shunts are all closed in very soon, usually immediately and within next 4 weeks, max. Because otherwise it is incompatible to life. So the best way to is, so can we hold on just a second? Yeah. So what happens to these shunts? That is sometimes asked in exam, and these are written in red fonts. So the umbilical arteries become the round ligament of bladder. This is how you will find in adult animals. The umbilical vein becomes ligamentum teres, sometimes also called the round ligament of the liver. The ductus venosus becomes ligamentum venosum. Ductus arteriosus becomes ligamentum arteriosum. And foramen ovale, after it is closed, it becomes fossa ovalis. These are anatomical structures you will find in adult animals, which are coming because of embryological structures which have digressed and not functional anymore. This has been asked many, many times in the past as in the exam. One final thing, the ductus arteriosus, we have talked about this ductus arteriosus over here. I will start the laser pointer. This ductus arteriosus, it closes after the baby is born, but sometimes it can stay open. That kind of situation is called patent ductus arteriosus, PDA, which is incompatible to life. Because when a baby is born and the heart is now complete with the separation of oxygenated and deoxygenated blood, this connection cannot be there. Because now you are mixing deoxygenated with oxygenated blood, which is not allowed. So this ductus arteriosus needs to shut down. And it is usually, there are a lot of molecular mechanisms now identified, but the prostaglandins and prostacyclins, this kind of molecules take a part in it. So you have to remember in pregnant animals, what kind of medication you are giving, because some of the medications, for example, ibuprofen or prostaglandin inhibitors, if you use them, they might keep the ductus arteriosus open, which is not allowed, because then in adult animals, most of the advanced vertebrates, including humans, mixing of oxygenated and deoxygenated blood is not allowed, because that is incompatible to life. Because we don't have hemoglobin F, we have hemoglobin A. And the hemoglobin F, which has done all this molecular magic, it's a magician. Moment the baby is delivered, the hemoglobin F starts disappearing and it is gradually replaced by hemoglobin A, which means hemoglobin adult, which we all have now in our blood. So this is a highly complex and synchronized mechanism Uh, Listen, it's difficult. There are no easy solutions to it, but it's also super exciting because a lot of things are going on. A lot of things are getting, the aortic arches are getting disappeared or connections. Don't ask me how this all happens. I can only show you what happens, how, why. It's an incredible structure. There are too many unanswered questions. We don't know. And today, because of the time crunch, we have just been on the surface. It's very interesting to know how the sinus venosus and the three venous systems, the cardinal veins, the umbilical veins, and the vitelline veins, how they give develop different veins, including the vena cava, and very importantly, the portal system. Those are very important parts of cardiovascular system. I hope you will agree with me. Then how the lymphatic system develops, how the different blood vessels of gut develops, how the celiac trunk develops, how the cranial mesenteric artery develops, how the caudal mesenteric artery develops. It's a really extensive discussion, which unfortunately I cannot go on today. I think it's a short, compressed 1 hour or 1 hour 50 minute kind of a lecture. At least I have tried to show you some of the complexities, what happens, some of the incredible machinery or molecular mechanisms that we often don't know that takes part into it, and the complex system of heart and the aorta and this septum. This is happening in different ways, and it usually happens very well. That is why we are all alive. Sometimes it does not. It happens. Again, those are called, what are the problems or issues that can happen due to complex or defective development? Unfortunately, I don't have time to go through all this because then we will be stuck here for next 5, 6 hours, which neither you have time for nor I. So I hope I will be ending over here. Maybe the moderator can take over if there are some time for questions and answers. Hello.
Okay, Doc.
Hello, Prof. Suraf.
Hello. Yes.
Yeah, thank you very much for your explanation about this topic. Is very interesting and nice presentation. Okay, all student to we will go to next session Q&A, Prof.
Yes.
And answer. Okay, do you have any question for this topic? Okay, please.
By the way, you can always send me emails. It's not to be always here. If you remember or if you have a question, you can always ask me later. You just drop me an email.
Good afternoon, Professor.
Yes.
My name is Gianoven Muhammad Yusuf.
Yes.
I have a question. Is there an embryological basis for the high prevalence of mitral valve disease in small breeds? Specifically, could there be a subtle defect in the epithelial mesenchymal transition during the formation of the endocardial cushions? Maybe that's all. Thank you for your attention.
So you have to repeat your question once more. I could not hear you very well. Can you repeat it, please, once more?
Is there an embryological basis for the high prevalence of MMVD in small breeds? Specifically, could there be a subtle defect in the epithelial mesenchymal transition or EMT during the formation of the endocardial cushions?
Yeah, the answer to is there is always an embryological reason. And one of the major thing is what I have shown you here, that is mostly what are the structures looking like or developing, what are the septum coming or the endocardial cushions are coming, but those are external structures. What I could not show you, because that is typically not covered in gross anatomy lectures, there are a lot of things happening internally which we are not seeing in our eyes. And one of those things is typically genetical factors. All these muscles developing, this happening. There are a lot of complex interplay which we simply don't know at this stage, or we have just started knowing them. One of the few, for example, different genes are getting activated by this time. And these genes are not only getting activated, they are getting activated in different parts of the body, in different segments. So yes, I'm pretty sure there are embryological basis, and it depends on the structure and some of the genetic factors that are definitely playing a role in all these small breed dogs. So my answer to you is, I'm quite certain there are embryological issues, and there are definitely, my pretty much guess will be, there are a lot of genes coming into the facts. And also the typical structure of heart that we have shown, the 4-chambered heart that we know of, that has a lot of species variation. And for example, in small breeds, the cardiac structure is also very different, so it fluctuates. As a result, I'm pretty sure there are embryological basis to all this, what are kind of different valvular diseases that you are naming for. Okay?
Thank you, Professor. It's
My pleasure. Thank you. You can always send me email if you have any further question.
Yeah. Any else? Yeah, please.
Okay, good afternoon, Professor Surav,
Thank
and
you.
thank you for the opportunities that Professor give me to asking about one questions. Because as far as I know, that not all of animal species develop 4 chambers in their cardiac. For example, like
Yes.
only have 2 chambers, and we only have 3
Yes.
chambers, and people generally have 3 chambers, but some of crocodile species have 4 chambers, but still with undeveloped septum.
Yes.
There any change that kind of animal have genetic disease or increased risk to get a cardiac disease or have an abnormal functions of the cardiac that causes by the situations?
Listen, what I have shown you today is a typical generic structure. Okay? You mentioned about crocodiles, right? Crocodiles is an amazing animal. And crocodilian heart is also super incredible. So this septum that I have shown you is pretty much not there. I would not say not there, but it is not like the way we are dealing with in our lives. What I have typically shown you is the architecture of the anatomy which is present in animal heart that is mostly around us and domesticated. The key point here is crocodiles can survive, or some other animals can also survive with not a partition but impartitioned heart, where the septum are either not there, or they are not fully developed, or they are not fully separative. They all have incredible physiology, and they all have different hemoglobin systems. We don't understand always, or we don't appreciate enough the hemoglobin systems that these all different species have. And crocodiles typically, of course, the reptilian systems, they are also very, very different. I don't have time today to go through all them. First of all, that is mostly discussed about zoology. It's a zoological topic more than veterinary, probably. But it's definitely true. These animals have, what I have shown you today is kind of what you will face in as such typical veterinary clinic. But there are many, many variations, and they all are fine. They go through it, they survive. And some of the diseases can definitely have these developmental issues. For example, I could not mention today because of time issue. Maybe you guys have already heard there are tetralogy of Fallot. Have you heard of tetralogy of Fallot? Anyone? Anyone? I don't know. Tetralogy of Fallot. Now, what happens is many of these genetic or developmental disorders in the cardiovascular system, and mostly in heart, of course, they are not only coming as one disease, there can be multiple issues. For example, this tetralogy of Fallot that I said, there can be a combination of pulmonary stenosis, ventricular septal defect, etc., etc., etc. So my answer to you, if I understand your question well, is that different animals typically have developed their own systems over evolution, And the structures that I have discussed today does not cover them all. It only covers typically veterinary subjects, for example, dogs, cats, etc. But if you go to the real animal kingdom, the different species variation of cardiovascular system, especially the partitioning of heart, is incredibly diverse. Second, some of these systems can go through a lot of issues, which can create a lot of diseases. Unfortunately, I could not discuss it today because then it will take a lot of more time. If Armanda and Tagita and other people of your faculty give me some time in future, or maybe when I am visiting, we can go through into a lot of details, which unfortunately I could not do today. However, you are all very, very welcome to send me emails. You have my email. If not, please get it. It's very easy. And send me an email, and I will be very, very happy to respond to the rest of my academic capability. Is it okay now?
Okay, thank you, Professor.
Thank you. Thank you. Any further question?
Good afternoon, Professor Suraf. Let me
Yes, thank
introduce
you.
myself. My name is Laras, one of the first years in veterinary medicine. So
Mhm.
I'm here to explain my thoughts and my questions. As
Yes.
far as I understand, some congenital cardiac pathologies may require medical therapy. For example, conditions like ventricular septal defects that need supportive treatment to manage cardiac workload. Therefore, as veterinarians, what factors should we carefully consider before prescribing medications during the fetal or embryonic stage to minimize risk to development? Maybe that's my question. Thank you.
Now listen, I am not a veterinarian myself. So the best thing to ask is, when you go to the final years, I think these things will be taught. What I can definitely tell you, that a lot of medications are there that interfere. For example, painkillers, one of the examples. Let's say prostaglandin inhibitors, prostacyclin inhibitors. There are certain drugs which you cannot provide or cannot administer during pregnancy. Doesn't matter which animal we are talking of. It can be humans, it can be different animals, doesn't matter. Because you have to understand this entire system of an embryo that is developing. It's a very highly metabolically active organism, or species, because it's developing a lot of organs. A lot of things are happening. We have only discussed certain sections of cardiovascular system today, and I think we have already seen so much is going on. And there are other systems in embryo. So there is too much going on. It's a very metabolically hyperactive space. And any medication you are putting in a pregnant animal, you have to understand that medication will ultimately reach the fetal unit through the blood, mostly by blood. So in a situation like this, there has to be proper maintaining of drugs which can be used and cannot be used during pregnancy. And I'm quite certain there is an extensive list of those kind of drugs available in veterinary literature as well. It is available in human literature, it is available in veterinary literature, doesn't matter. You have to be very cautious when you are putting drugs inside a pregnant animal or pregnant human, doesn't matter. Because all these drugs, they're basically fundamentally chemical molecules, and they can interfere with a lot of things. For example, like you have already rightly mentioned, the ventricular septal defect. We have seen how the ventricular septum develops. It's a very complex process. A lot of signaling mechanism is going on. And your drug, the drug you have put inside as a veterinarian, if it interferes with it, the ventricular septum defect will either not develop, or even if it develops, it will be ineffective or defective. So there is a load when you are putting any medication in a vertebrate species in pregnancy. There is extensive literature, and I'm quite certain those kind of literature, what kind of drugs to be avoided, are also available in veterinary literature. You can easily search it. If not, if you come back to me, I can find it from my colleagues and send it back to you. Okay? But it is a very important question. It's a good question because the drugs that you are putting to your patient, if it is a pregnant patient, it can interfere with the development. It can cause issues with cardiac development, including ventricular septal defect, and not only ventricular septal defect. What I did not touch today or did not have time today, we talked only about the heart as an organ. But I had to skip that heart is a pumping organ. So there are different nodes, electric nodes that kind of create that pumping mechanism, otherwise called as systole and diastole. So these are the different nodes. How? Because heart is an electrically stimulated organ. It continuously pumps blood. How this pumping system develops. Unfortunately, I could not make it today. So these are very exciting discussions. Hopefully, I can meet you sometime in the end of the year, and then we can go through it if you are interested. So what I have actually given it today, if you look into the title of the talk, it's called Glimpses of the Development of Cardiovascular System. It's only glimpses. Okay. Hello.
Okay.
Thank you. Listen, guys, you can always send me email. Absolutely fine with that.
Okay. Okay.
Okay then. Any other question?
Any more question? One more again. Last one. Have you understood what topic today? About embryology. Okay. One else? Yeah, please. One more again, Prof.
No problem. Welcome.
Thank you so much. Yang Laras.
Hello, good afternoon, Professor Suraf. Thank you for the opportunities. And let me introduce myself. My name is Rosa Fitra Alam, and I would like to ask some question about embryology of cardiovascular. Why is the process of cardiac looping crucial in early heart development? And is there any consequences if looping occurs in the wrong direction? Thank you.
It's a very interesting question. So what she has asked is, we have talked about looping. I have to share the screen again. It's a very interesting question. Let us go back. We unfortunately or fortunately we have to go back. Hold on. Why is it not happening? Hold on. Just a second. Yeah. So I have to close it. So it's a very interesting question. So we were talking a lot about looping. We get back here, isn't it? Sorry. Just a second. Just a second, guys. You see it, right? You see this, isn't it?
Yes, Professor.
Good. So this looping you are talking about, right? Is there be a problem? I think yes. Because this looping, if you can see my cursor, gives development to certain anatomy. And the repercussion of this looping is it puts the ventricle on the ventral side and the venous system, that is the atria, on the dorsal side. This is how heart has developed. If there is no looping, number one, these two endocardial tubes will struggle to fuse. So the looping happens. First of all, this looping brings these two cardiac tubes close by. So that is number one. So when the looping happens, these two cardiac tubes come close by and form that common tube. So no looping means they will not come close, number one. Number two, the looping also means because of this folding, the ventricle comes towards the ventral side and the venous part, that is the atrium, comes to the dorsal side. That keeps the blood flow the way it is. If there is no looping, this folding and this coming of two endocardial tubes will struggle, and that will upset the entire architecture of anatomy that we have in the cardiovascular system, including the heart. If I ask all of you which side the heart is tilted, you will say it's on the left. This entire anatomy, the way vertebrate systems have developed, will be upset if the looping is not happening properly. The next question you have asked is, if this is not happening, can the looping happen other way? The answer is yes. There are cases, so we have seen the primitive ventricle becomes the left ventricle, and the proximal 1/3 of the bulbous cordis becomes the right ventricle, and it tilts towards left. That is why we typically say our heart is towards the left. However, there are exceptions to it, and the looping, if it does not go well or it does not go the way it should have been, sometimes the looping, because of defective looping, the left ventricle can tilt towards the right. We still don't understand why it happens because there are a lot of genetic factors coming to it. But there are cases, and I have seen those cases in my life, where the heart is actually because run towards right. These type of cases are typically called as dextrocardia. That means the heart is typically facing right. Is it a problem? The answer is often no. I have seen many cases where the heart is, not many, but few at least, the heart is towards the right, especially in human patients, and they did not even notice. They did not know. It is typically discovered accidentally because the patient is otherwise fine. So, is it a problem? Physiologically, often not. Sometimes, but often it is not a problem. The patient can survive very well. In fact, it's not even a patient because the subject is not sick. It can happen because of this looping. The dextrocardia can happen. There are cases where not only the heart, but everything, there is a complete site reversal. So not only that, for example, liver is also on the right, but on those cases, everything is site reversed. So the heart is towards left, the liver is towards left, heart is towards right, liver is to the left, spleen has gone to the right. So everything gets reversed. And those type of cases are called situs reversus. So is it a problem? Often not. But because of these issues, problematic looping or incomplete looping, there can be site reversal as well. And in few cases, there can be issues as well. For example, we were talking about ventricular septal defect, etc., etc. But as long as the compartmentalization is okay with the species, they are often asymptomatic. So technically, there is not a problem to survive. In some cases, there can be. So it's a complex question. There is no clear-cut answer, but looping is very important for heart, and it's better goes that way rather than any other chances, because looping is what gives heart the structure that we know of. No looping means no structure of heart. It's just a simple tube. And the further developments will not happen. For example, the aorticopulmonary septum will not develop. It will create a mess. There are chances. Fortunately, it does not happen often. Patients are often asymptomatic, but it can happen as well. So there can, but yeah, I would prefer it goes the way it is rather than any other exceptions. Okay.
Okay, thank
Hello.
you for the explanation, Professor.
Thank you. Thank you.
Any more question? You know? Okay. No more question, Prof. I think.
Thank you.
Yeah. Thank you very much for your explanation. This topic.
No, listen. One thing I would like to say before I part off. It's a complex thing, and it's a 3D thing. It's everything is happening in 3D. Now, it is difficult to show everything by sharing a 2D screen on Zoom.
Yeah.
Because it's a complex thing. A lot of things are going on, lot of things. And when I was taught by my teachers, we always ended up in a blackboard with chalks. We used to draw, we used to delete. Otherwise, the concept is very difficult. So I completely understand if you don't have grasp over everything. It's just a flavor of it. There are too many complex stuff. I have left, at least I would even say I have left 60% because yeah, it needs complete days after days to understand the development of cardiovascular system. For example, we have talked about heart. I told you I could not discuss the different nodes that create that pulsation. I could not discuss with you the different venous system that develops, how the portal system develops, how the superior and inferior vena cava develops. I could not discuss with you how the different other systems develop. And finally, I could not discuss with you how the lymphatic system develops, which is also part of the cardiovascular system. So your adult vertebrate body is super, super complex. You can always email me. I will try to clarify your doubts. At the same time, when I'm in Indonesia, hopefully end of this year or beginning of next year, if you guys are available, I'm absolutely face-to-face, day-on intensive courses. And I think these are amazing, incredible stuff, which is so interesting. And we can keep discussing for hours and hours. That's all from me for today.
So much for suggestion and your opportunity for me and my student
My
today.
pleasure. Yes, my pleasure.
Okay. Before we close this class, we take a picture together with the
Absolutely.
yeah.
I don't know, but listen, can I take a screenshot and share in my Twitter? Is it okay?
Yeah.
Absolutely. So I'm doing a print screen now.
Yeah. Okay.
Yeah. So yeah. Oh no. Hold on. Yeah. Print screen.
Yeah.
Yeah. All done. Hold on. Because I will share in my Twitter. I'm very active in social media, and I think it will be nice. Hold on. I know it has not worked out. I don't know why, but just a second. Rectangle. Yes, I have taken it and I will share with it. And listen, it's great to meet you guys. What shall I say? I'm looking forward to meet you all. I
Yeah.
love Indonesia. The people are nice. The weather is far better than Ireland, for sure. It's
Yeah.
very cold out here. So yeah, life is probably better sometimes in there.
Yeah.
And I'm definitely going back. I will be holding, I think, workshop and other things. Plus, I have to go to some other places. I keep on joking with other guys I know in UNPAD that they keep on telling me you should go and visit Borneo. And I keep telling them I'm very scared of snakes. So Borneo has so many snakes. So I'm very, very scared of it. So, but other than that, listen, guys, I'm looking forward to meet you in Indonesia.
Yeah.
And there is so much to discuss.
Yeah.
And we can sit down and keep talking forever. No problem.
Yeah. I hope so. Thank you so much.
Email.
See you.
See you. Bye. Have a nice day. Bye. Bye. Okay.
Have a nice day.
Same to you guys. Bye.
Bye. Bye.